Hi, my name is Najah binti Muzaffar, and today I will be presenting my final year project titled The Potential of Clitoria Trinity Crude Extract for Microbial Growth Assessment. Before we dive deeper into my project, let's see if any of you recognizes this flower. Still no clue? Think about what they're used for instead in our country. Yep, that's right, butterfly pea flower were used in many of our cuisines, including nasi kerabu and our traditional kuihs. Now, there's something this flower could also do. If you guess that they can change color when exposed to acidic things, you're correct. These flowers are able to change color in response to various pH levels due to their special pigment called anthocyanins. And these, my friend, are what my project revolves around. But that's not all. In 2019, Deja Taylor invented color-changing medical sutures that indicate wound infection using beetroots. Bacteria releases byproducts that are sometimes acidic and alkalic, as seen in wound infections. Therefore, her sutures aim to be a cost-effective color-changing infection indicator in real-life application. So, this gives me an idea. Why not use the same theme but use a plant that can easily be grown and found in Malaysia? So that's what I did with butterfly pea flower, or scientifically known as Clitoria trinity flower. My study had two main objectives. Firstly, to evaluate the pH sensitivity of Clitoria trinity extract by observing its colorimetric changes. Second is to determine the pH values of microbiome metabolic byproducts during different bacterial growth period. Let's see what approaches I did for my project. The process began with acquiring dried butterfly pea flowers. Then, the petals were ground into powder using a blender and mixed with distilled water, then heated to obtain a crude extract. Next, buffer solutions from pH 1 to pH 14 were prepared using hydrochloric acid and sodium hydroxide. Then, a few drops of previous flower extract were added to each buffer solution to observe color changes. This is to prove the abilities of the flower to change color. For microbial testing, E. coli and SRS were inoculated into a prepared nutrient broth and then incubated for 24 hours. Next, small amounts of the bacterial samples were taken at each growth phase, then mixed with 5 drops of the previous flower extract, and the color change was observed. Now, let's look at the results of my research project. As you can see, in buffer-only solution, the extract showed clear pH-dependent color changes, where the color changes from red and purple in acidic conditions to green and yellow in alkaline. This is due to the deprotonation and degradation of the anthocyanin pigment. At low pH, the pigment forms the red flavillum cation. As pH rises towards neutral, it shifts to the purple and blue quinoidal base, and in alkaline conditions, degradation produces carbonyl pseudobase and chalcone, turning the pigment green to yellow. Moving on to the next results on colorimetric changes in Clitoria trinity extract across different bacterial life phases, we can see that in the lag phase, there was minimal activity and no color change. Therefore, the color remained dark blue. In the log phase, the bacteria actively metabolizes nutrients, thus producing slight acidic byproducts like acetic acid. This causes a lighter blue color change in the pigment. Next, in the stationary phase, alkaline compounds were released due to the metabolic transition by the bacteria, where it digests the acidic byproducts and releases alkaline compound instead, such as ammonia, turning extract dark green. Finally, in the death phase, cell lysis occurred, releasing more basic contents, shifting the color to light green or greenish yellow. So, from the results I've achieved, in conclusion, Clitoria trinity extract showed consistent color changes across pH levels and successfully detected microbial growth phases, acknowledging the success of both objectives. This proves that Clitoria trinity flower extract is a viable, eco-friendly alternative to synthetic pH indicators and can be applied in a low-resource settings for microbiome monitoring. Before I end the video, just as Deja Taylor demonstrated with her color-changing sutures, my research on Clitoria trinity extract shows the potential of natural pigments as effective pH indicators. If we harness its color-changing properties and incorporate them into medical tools such as sutures or bandages, we could enable early infection detection in a simple, cost-effective way. This innovation could not only save lives, but also reduce medical costs, making advanced healthcare more accessible to everyone. That is all from me for my final year research project. Thanks for watching and goodbye!